Hello everyone, today we're going to talk about integrative viral vectors, AAV. Gene therapy is a medical technique that aims to treat or prevent disease by changing a person's genetic material. This can be done by adding a healthy gene, correcting a faulty one, or regulate gene expression. The main goal is to treat the root cause of the disease rather than only its symptoms. To deliver genetic material into cells, science uses viral vectors. These are viruses that have been genetically modified so they can no longer cause disease, but still maintain their natural ability to enter cells efficiently. In this way, the virus becomes a delivery vehicle that carries therapeutic DNA into the target cells. The adeno-associated virus, or AAV, is one of the most widely used viral vectors in gene therapy. It is a small, non-enveloped virus from the parvovirus family. AAV cannot reproduce on its own. It needs a helper virus such as adenovirus or herpes virus, which makes it non-pathogenic and very safe for humans. AAV has an inconsiderable capsid about 20-25 nanometers in diameter and contains a single-strand DNA genome of around 4.7 kilobases. It can infect both divine and non-divine cells and provides long-term and stable gene expression, which is essential for therapeutic uses. AAV offers several important advantages as a gene delivery vector. Low immunogenicity, it causes only a mild immune response. High safety, it does not integrate randomly into the human genome, minimizing mutation risks. Broad toppings, it can target many different tissues and cell types. These characteristics make AAV a safe, versatile, and effective tool for gene therapy applications.
The adeno-associated virus, or AAV, is natural unable to replicate on its own. It needs a helper virus like adenovirus to transform into a safe therapeutic vector. Saving remove the viral genes responsible for replication and capsid formation, known as rep and cap. They keep only to a small sequencing called ITRS, or inverted terminal repeats, which are essential for replication and packing. In the remaining space, the therapeutic gene is inserted along with a promoter and other regulation elements that control how, when, and where the gene is expressed. Once designed, the vector must be produced in the laboratory. This is done in HEK293 cells, using a system of three plasmids. The first plasmid, called the transfer plasmid, carries the therapeutic gene flagged by the ITRS. The second plasmid proves the rep and cap genes, which allow the virus to assemble its potential. The three plasmid, this helper plasmid, provide the additional viral function required for replication. When these plasmids are introduced into the cells, they work together to produce millions of AAV particles. Afterwards, these particles are purified to separate those containing the therapeutic DNA from the empty ones, resulting in the clean and effective viral vector ready for medical use. Once designed, the vector must be produced in the laboratory. This is done in HEK293 cells, using a system of three plasmids. The first plasmid, called the transfer plasmid, carries the therapeutic gene flagged by the ITRS. The second plasmid proved the rep and capped gens, which allow the virus to assemble its potential. The three plasmid, this helper plasmid, provide the additional viral function required for replication. When these plasmids are introduced into the cells, they work together to produce millions of AAV particles. Afterwards, these particles are purified to separate those containing the therapeutic DNA from the empty ones, resulting in the clean and effective viral vector ready for medical use.
In gene therapy, AAV works at the molecular and cellular level to fix defective genes. The virus carries a functional DNA sequence that replaces or supplements the patient's faulty gene inside the cell nucleus. Once inside, the new DNA remains as an episome not integrated into the chromosomes and is transcribed into messenger RNA, which the cell uses to produce the missing or defective protein. For example, in hemophilia, liver cells receive the coded gene that encodes the chlorine factor protein, restoring the normal biochemical pathway of blood coagulation. In muscular dystrophy, AAV delivers a shortened but functional version of the dystrophin gene to the muscle fibers, allowing synthesis of dystrophin and improving the muscle contractions. In inherited blindness, AV introduced the healthy RPA65 into the retinal cells, restoring the enzyme activity needed for the visual cycle and allowing photoreceptor cells to respond the properly to light. Honda-approved therapies is Zolgensma is used to treat spinal muscular atrophy SMA, a deadly disorder where mutation in the SMN1 gene cause motor neurons to die, leading to paralysis. This therapy uses AAV9, which can cross the blood-brain barrier to deliver a healthy copy of SMN1 to motor neuron after a single intravenous injection. Once expressed, the new SMN protein protects the neurons and restores the motor function. So AAV vectors can be delivered in different ways depending on the disease and the target tissue, right? They can be given intravenously to raise the liver, heart, or nervous system intramuscularly for muscle disease, or injected directly into the specific organs such as the eye or brain. The flexibility allows AAV deliver genes precisely where they are needed because AAV is safe. So produce long-term gene expression at target cell efficiently. It has become one of the most powerful and reliable tools for treating genetics disorders today.
Despite their success, AAV vectors have some limitations. Their main disadvantage is their low gene carrying capacity, as they can only transport gene up to about 4.7 kb. Immune responses can also be generated against the capsid, which hinders its reuse in the same patient. Furthermore, the high production cost limits its availability. Regarding safety or the integration into genome is rare. It aspect that is monitored to prevent a wanting. The future of AAV lies is developing modified or synthetic capsids that improve their specificity and reduce the immune response, as well as combine gene editing and readministration strategies. In short, AAV has transformed gene therapy, but it still faces technical, economic, and ethical challenges that must be addressed to make it accessible to more patients.